In this second of two videos looking at genetic information for a GOB chemistry class, we left things off in video one just by recapping on the dual role of DNA as the messenger in a bottle through time. The goal of DNA is to not only replicate itself, but to replicate protein. Why replicate itself? Because you need lots of copies of DNA to make enough protein. And once you have enough copies of DNA, you can start to make protein. So let's, in this video, start to look at how that actually happens. It happens via RNA, which is the ancestral precursor to DNA. So remember, RNA is ribonucleic acid. It is typically a lot shorter than DNA. It's chemically less stable than DNA because it's oxygenated on the 2' carbon of the Haworth projected ribose unit. So if you go back to the first video on this topic, then you can look at the structure of a nucleotide, and it has a deoxygenated carbon in its nucleotide. Anyway, we can see here that there are 3 flavors of RNA. There is ribosomal RNA, that's what the R stands for. There is messenger RNA, that's what the M is. And there is transfer RNA, that's what the T is. Most of the RNA is actually ribosomal RNA, and ribosomal RNA essentially is where let's just say it's where site of formation of primary protein structure. So if you go back to my video on the primary structure of protein, so the amide bonds or the peptide bonds if you're a biologist, then that's the primary structure based on which alpha amino acids are laid out. Well, the ribosomal RNA is essentially like a clipping machine. It's what clips together the fragments of RNA. Sorry, the fragments of alpha amino acids to form the protein. Okay, but who tells the ribosome who lays out the structure of alpha amino acids anyway? You know, there are 20 natural alpha amino acids, and they're coming in thick and fast. Who tells them? Who choreographs this flow of chemical information? Well, that's the messenger RNA that tells what's next, and then the transfer RNA is what actually chaperones the alpha amino acids. So essentially, we've got the information from the messenger or the message. We've got the chaperone of the message, the transfer, and then we've got the ribosome that acts upon the message. The information is faithfully communicated via base pairing. So this is, again, base pairing. But it's not the base pairing. In fact, maybe I shouldn't call it base pairing. I don't mean base pairing as in ATGC. So I probably shouldn't say that. I should probably just say hydrogen bonding. So it's analogous to base pairing in as far as it's hydrogen bonding. But we'll take a look at it in the next couple of slides. Okay. So here's our alpha amino acid. here's our alpha amino acid. We've got the amine part. Let's make this slide a little bit bigger. We've got the amine part here. It looks like it's in the Zvita ionic form. Lots of good vocab from a previous video here. This is the physiological form of the alpha amino acid at its isoelectric point. The amine part, we've got single nitrogen-hydrogen bonds. We've got the alpha carbon here with its functional group here, R. And then we've got the carboxylic acid part here with its carbonyl and its dehydrogen. It's lost its acidic hydrogen here. So amine acid, amino acid, sandwich in the alpha carbon that is loaded with its functional group here. Okay, the amino acid is actually tethered or chaperoned on the transfer RNA. So the transfer RNA chaperones or is a chaperone to is a chaperone of the alpha amino acid. So the alpha amino acid is the payload, but you can call for a specific alpha amino acid by calling for a specific tRNA molecule. And we need to look at how you do that with the codons and the anticodons. But essentially you want an alpha amino acid, a specific one, you call for a specific tRNA molecule. So that's kind of nifty. So how does it work? Well, we have the codon table here. So at least for my students, I would provide my students with the codon table. You should consult your class to see if you would be provided with this also. I think it would be more common than not to be provided. So let's see how we navigate this. So we've called out for our tRNA. How do we call out? Well, the mRNA, the messenger, comes from the DNA, right? So the DNA then calls out the message. The messenger RNA goes out and spreads the message. And the message it actually spreads is a codon message. A codon just means a code. So here's my code, and my code is UCA. My code is UCA. My first one is U, my second one is C, my last one is A. Okay, so my first base is U, so let's go here. First base is U. My second base is C. And my third base is A. So my third base, so I'm going clockwise around this particular table. My third base is A. So if I home in, that tells me I have the alpha amino acid serine. So my code is coding for serine. But I need to call the chaperone, the specific chaperone that has serine. Well, the specific chaperone that has serine has the complementary base pairs. The anticodon is the opposite of the code. So this is the opposite code. Okay? Notice again the complementary base pairing. U goes with A, C goes with G, and A goes with U. So we have UCA complementarily base pairing with A, G, U. Notice we don't have any T. We don't have any thymine here because it's not DNA, it's RNA. So we have the presence of U instead. But it's this complementary base pairing that calls for the specific chaperone that has the specific payload serine. So it's all managed via hydrogen bonding. This is a great overview of how it all works. So, and I would encourage my students to be able to go up and down this diagram. So here's our DNA. We've got the double alpha helix of DNA. So we've got the 5' and the 3' complementarily interacting via hydrogen bonds. So here are the hydrogen bonds here. A goes with T, T goes with A, G goes with C, A goes with T, T goes with A, G goes with C, etc. So we've got the information. So let's just tidy this up and let's look at how the information is relayed. So this is the relay of the genetic information. So let's just look at one of these and then we can do it for all of them. Let's say the information is ATG. Then that information is protected in a double helix of DNA via the complementary partners of AT and G, which are T, A, and C. Let's say I'm ready to transcribe that information. So I would transcribe that information into the message. The message comes via the messenger RNA. So notice that I faithfully reproduced the information, my information, ATG, but I've got it in RNA language. So I've got the A, I've got the G, but remember there's no thymine in RNA. So I've converted the thymine into uracil, but otherwise I've still got faithfully the same information. I've transcribed the information via transcription. Likewise, I don't have thymine, I've got UU, but otherwise I've gone via transcription, I faithfully reproduced the information. Okay. The information here is the codon. So these are the codons. Codon is a cluster of 3 bases. This is what my table has in it, my codon table. So my codon table is the RNA version of my DNA information. So let's just look at this first one here. AUG, that calls out for the anticodon, which is the complementary base pairing. So the anticodon comes from the chaperone, the transfer RNA, which is transferring the alpha amino acid. So these proteins, these alpha amino acid pieces, are chaperoned via the anticodons. So UAC chaperones methionine, AAC chaperones leucine, CCU chaperones glycine, etc. And it's all mediated via this hydrogen bonding between complementary base pairs, purines and pyrimidines, purines and pyrimidines. And then ultimately, we have alpha amino acids, so these are all the alpha amino acids, building blocks of our protein. Here's our peptide bond, or if you're a chemist, it's an amide bond. And we've essentially laid down, with the help of our ribosomal RNA, so we've got our rRNA here, which allows us to make this protein. We've essentially got our primary structure of our protein. This can then go on to secondary, can then go on to tertiary, and if necessary can go on to quaternary in the hierarchy of protein structure. But it all starts with the primary structure, which came from the DNA. So that's the message in the bottle, and it's all mediated via hydrogen bonds. Okay, so again, I would definitely require and recommend for everyone that you can navigate up and down this structure from the DNA through the workers, who are the RNA molecules, all the way to the final product, which is the primary structure of protein. That's what's supposed to happen, but things can go wrong, and sometimes things going wrong is intentional and sometimes it's not. Let's look what could go wrong. So if things go wrong, it's called a mutation. Mutations can be disastrous, they can be benign, they can lead to disease like cancer, they can lead to evolutionary advantages, but they're all mutations. A mutation is anything that alters the nucleotide sequence of DNA. So remember, these are the bricks in our wall. So it alters the structure of the wall. A mutagen causes mutation, and mutations can, although not always, they can lead to genetic disease. So this is essentially a vocab slide. Let's have a quick look at different types, and this is actually our last slide, so this will be a much shorter video, which is always merciful. So there are 3 types of mutation pointed out on the slide, and they're not all equal in the severity. So let's see if we can tease out which, if you had to have one, which would be best. So let's say this is the, and I'll actually zoom back in. Let's say this is the normal case here. So we've got normal here, so this is what was intended. We've got complementary base pairing. So this is the template strand of the DNA. So the informational strand would be above it. The information goes to the template goes to the message. Okay. Okay, so this is the template here. So we've got base pairing between A and U because there's no thymine in RNA, so we're in RNA language. So A always goes with U, A goes with U, T goes with A, G goes with C, C goes with G, etc. That's normal. The message, remember, this is the codon, clusters of 3 base pairs, and this particular codon codes for this particular alpha amino acid, which is phenylalanine or phenylalanine. This particular codon is, I'm looking at my codon, my list here. This is tyrosine. So we've got phenylalanine, tyrosine, we've got GLU, glutamic acid, and glutamic acid. Okay, so let's say that's normal. Now, that's great. From a chemist's point of view, if you had access to the actual structures, which you would on an assessment, you would know if you interpret the structure of phenylalanine, the functional group is hydrophobic. So it won't dissolve in water. Tyrosine is also, if I can locate it again, tyrosine is also hydrophobic. And glutamic acid is hydrophilic. So that means it will dissolve in water. And obviously, again, it's hydrophilic. So let's consider the solvation properties, whether or not you're hydrophilic or hydrophobic. Okay, let's look at a substitution mutation. So let's see what happened here. So we're looking at this area here. So all that's happened is instead of cytosine, we've substituted, somehow cytosine has been substituted with thymine. And instead of cytosine calling out for guanine, it was substituted to thymine, which calls out for adenine. Now that's great, but what will it mean for the alpha amino acid? Well, instead of GAA, we've now got AAA. So instead of calling out for the alpha amino acid glutamic acid, which is hydrophilic, it's now calling out for the alpha amino acid lysine. And lysine, if I can see the structure real quick, lysine is hydrophilic. So lysine is hydrophilic. Now it's not acidic, so it's a basic structure. So glutamic acid obviously is acidic. So they're both hydrophilic, but lysine is basic. So we've kept the same solubility properties in our substitution here, but we've changed the type of chemistry that solvates from an acidic alpha amino acid to a basic alpha amino acid. That could have consequences. Okay, so that's one way as a chemist we would interpret that mutation. So just a substitution of one of the bases in the DNA strand. What about an insertion? So I would say the substitution is the smallest impact, let's say. Insertion, here we've inserted an extra base. So if we compare with the normal, we didn't replace anything, we just added an extra cytosine, which essentially pushed everything along. So the original CTT was pushed along here. So the final T of this DNA, ultimately of what would have become this codon, got pushed onto the adjacent one. And we've now got a brand new, so it essentially is a domino effect. We've inserted an extra base pair here, which has totally changed not only this codon, and therefore this alpha amino acid, but every one thereafter. So this would be a larger, or it has a larger impact. So potentially much more devastating. Almost certainly it will be devastating. Okay. Almost certainly it will be devastating. So let's have a look, at least at the immediate codon consequences. Okay, so by inserting that C and pushing everything along, we've now changed the codon to GGA, which now codes for the alpha amino acid glycine. Remember, glycine was the only achiral alpha amino acid because the functional group is hydrogen. So the alpha carbon is achiral. It's got no chirality center. Anyway, glycine, you recall, is hydrophobic. So just looking at this codon, because of its alteration, we've coded for a hydrophobic alpha amino acid. Now this is a great difference from a hydrophilic alpha amino acid. Arginine, ARG here, the alpha amino acid arginine, is hydrophilic. Just as this was also hydrophilic. So we've swapped out a hydrophobic for hydrophilic ad nauseum for the continuation of this particular gene. So this would be a devastating. So this is possibly benign, so no major devastation to the host, whereas this insertion mutation is probably a devastating mutation, which would lead to disease at the very least. Deletion is the third and final example of mutation in these slides. So instead of adding something, we've just taken something away. So comparing it with the normal one, which was right at the top. So maybe if I can make it smaller, I don't have to keep scrolling. So instead of CTT, we've erased, let's see how they represented it here. Yeah, they've just erased, actually, instead of here, we're looking at these three here. Instead of ATG, we've lost one of the A's and we've shuffled everything along. So we've got TGC as a brand new piece of template DNA. So you can have a look at that. That would be a deletion. Again, it's likely devastating because it affects everything. So I would say probably devastating. So I don't mean it will kill you instantly, but it would be devastating to normal health, however long that takes to manifest. So in this specific example, so let's see at I'm just interested in the ones that overlap here. So we've got lysine, which we said earlier in our substitution mutation is hydrophilic. And then arginine, which we said in our insertion mutation was hydrophilic also. And again, we're always trying to compare that with the original one, which was the normal case. Okay, so we have to, I guess, stand back and try and compare this with So we're essentially looking at, oh, didn't want to do that, did I? Maybe I can just go. Okay, so we're really just looking at these. Because that's how, these are what are in common. So that will conclude this video, the second of 2 videos on genetic replication for an introductory chemistry class.